Okay, hello everyone. It's great to be here today. I'm going to be talking about my project detecting interspecific positive selection using convolutional neural networks. And I think all of the talks that we've had today have been at the population level. And so we're going to switch things up a little bit and talk about evolution at the macro evolutionary scale. So the question that I'm looking at is, given an alignment of protein coding sequences from different species related through some evolutionary history, can we detect the presence of positive selection? And it's not a new question, but we're going to try answering it in a novel way with machine learning. So the way this has traditionally been done is that we will fit our data to a model that includes a parameter describing selection. And this is often this value omega, which is the ratio of rates of non-synonymous to synonymous substitutions. Non-synonymous substitutions being those in which the substitution leads to a change in the encoded amino acid, and synonymous substitutions don't lead to a change because of the redundancy in the genetic code. Most amino acid changes are thought to be deleterious, and so are assumed to be, in most cases, selected against. And synonymous changes are thought to be, where they don't result in an amino acid change, are thought to be or considered to be neutral. And so by this reasoning, we can interpret an omega of greater than 1 to be indicative of positive selection, where we have a higher rate of these non-synonymous mutations being fixated. And so in order to estimate omega, for our sequences, we need the associated tree, a multiple sequence alignment, some model of sequence evolution, and a statistical method. As for the model of sequence evolution, typically what is used is a codon substitution model, which is a Markov process describing the transition from one codon to another that can be achieved by a single substitution. And because we're working at the codon level, we have both the nucleotide and amino acid information, and so we can directly estimate omega on a per tree, per branch, or per codon site basis. As for implementing this, we can use maximum likelihood, which can be deployed through a range of software, but one of the most common is PAML. And yes, it uses maximum likelihood and it works very well, but it does have some limitations. One of which is that it can be overly conservative. So if you set a false discovery rate, actually the power of the method is less than you'd expect. And contrastly, in other cases, you often run into high false positive rates, and this is due often to alignment error, because there isn't any accounting for alignment error in the model. It's whatever the alignment is taken as truth, and to try to give some intuition for this. So I have some simulated sequences here, and on the bottom is the true alignment, and then above that is the sequences realigned with clustral. And up to kind of halfway in the alignment, they tend to agree, and it's looking pretty good. And then it gets to tricky regions with lots of indels, and we can see a lot of over alignment. And in this section here, what looks to be spurious mutations, which would inflate the omega value that's estimated. So what I'm asking is, can we improve upon these methods using convolutional neural networks? Or perhaps slightly more realistically, let's just see what happens when we throw convolutional neural networks at this problem as a proof of concept. So why convolutional neural networks for this task in the first place? So probably you're aware that CNNs have had a lot of success in image recognition tasks. And images have a grid-like structure where each point in the grid is a pixel. And multiple sequence alignments have a similar structure, but instead of a pixel, we have a nucleotide or gap representation. And in NCNNs through convolutional layers, both local relationships and higher order structures are detected to end up with the final prediction. So in this kind of toy example here, you have lower level features being picked up so that you can detect things like eyes and ears to make the final prediction. And you could imagine similar structures, not similar, but analogous structures being picked up in multiple sequence alignments, like signatures of positive selection, or even through misalignments, the kind of blurred motifs that you would pick up as quirks of the alignment algorithms themselves. Other reasons for using convolutional neural networks. So by virtue of being here, the machine learning methods being applied to evolutionary questions is becoming much more prevalent. But at least when this project was started, it wasn't really the case. But there were some applications in population genetics and tree inference. And although we feel that we're doing something novel here by applying it at the macro evolutionary scale, success in these related fields gave us hope that it could work here too. Potential advantages going into it that we thought we could see was the innate ability for feature detection, like I mentioned, above that, which is necessarily included in models. We could be able to detect features beyond that. And kind of in the same vein, use of the whole data set rather than just summary statistics, and also potentially making detection faster if we're scaling up the problem. So this led to the creation of Omega AI, which is referring to the final structure of a trained CNN that we have. Okay, so I'm just going to go through the workflow now. I think it's quite familiar to everyone, this kind of workflow now. So we start by simulating sequences, and we know that this is because we don't have any, or at least not much, data for which we know the ground truth about its selection status. And so by simulating, we always know whether the sequences have evolved under positive selection or not. And so we can label our data, and thus it becomes an example of supervised learning. And so we simulate sequences that have undergone positive selection. Oh, sorry, let me restart. So here I'm showing a color-coded alignment of what something we might simulate looks like. So each codon site in the simulated alignment will be assigned an omega value under which it will evolve. And sequences that are undergoing positive selection, they will have some sites which are allowed to evolve under an omega of greater than 1, and those which are not undergoing positive selection, all of their omega values will be less than or equal to 1. And for this problem, there's a huge parameter space under which we could have simulated sequences. But for our example, we chose to look at a constrained version of the problem. And so everything that you see is simulated under this 8-taxa symmetric tree. And we also simulate indels, which are the gray gaps that you can see in the alignment. We simulate 1 million trees or multiple sequence alignments for training, and then we have a smaller data set for testing afterwards. Okay, so even though we know the true alignment because we simulated the sequences, we realign them with an aligner. Here, pretty much always it's Clustal, because Clustal is fast and widely used. And we realign because in empirical data, you don't know the true alignment, so you have to align anyway. So it makes it slightly more realistic. The other part of it is that we actually want the neural network to be exposed to alignment errors, so hopefully it can learn how to deal with them. And so we convert the simulated nucleotide sequences into amino acid sequences, and we align them using Clustau and then convert back to nucleotide sequences. And we do this to conserve the reading frame. Once we have simulated and aligned our sequences, we then transform them into a one-hot encoded tensor, so that each nucleotide or gap character has a binary representation. Okay, so once we've done all that preparation to gather our training data, we can then train the neural network. And as I said, it's labeled data, so it's an example of supervised learning. There's 2.5-ish million trainable parameters and 6 convolutional layers. And probably most noteworthy is for the first convolution, the kernel has a width of 3 and a stride of 3 to try to encode this idea of a codon. And in the end, what we're trying to do is train the neural network to give a binary classification. So meaning that can it detect any single site in the whole alignment that has evolved under an omega of greater than 1. And once we've trained the neural network, we can then test it with our test data, obtain some classification metrics, and we also use that same test data and evaluate it with PAML to compare the two methods. And then once we've done that, we can then evaluate it and look at some results, which we're going to do now. Okay, so here I'm showing you results for what we've been referring to as our baseline parameters. And this is whereby the scaling of the simulation tree for every branch is scaled to 0.2 substitutions per codon site. And on the left here, we have an ROC curve, which is telling us about the trade-off between true positive and false positive rate as we vary the decision threshold for the two methods. And we have something similar but slightly different on the right, talking about the precision recall trade-off as we vary the decision threshold. The red dots, so on the bold line, which is representing omega AI, that red dot is a threshold of 0.5, which we used for training and we have just typically been using throughout. And then on the dashed line for PAML, the red dot is a significance level of 95%, which is commonly used in the literature. And so the data on the top, which I just showed you, that's for sequences aligned with Clustau, which were used for training, and then also sequences were aligned using Clustau for testing as well. And then we take that same test data set and align it with 3 different aligners, mathed and 2 versions of Prank. And we plot that below. I'm sorry, it's a little bit busy, but the main takeaway is that the bold lines are above the dashed lines, and so we're happy about that. Something of note here, I think, is that the bold lines representing evaluations by Omega AI on different aligners are quite close together, which says to me that the network is able to generalize quite well to different aligners. Okay, so as I said before, what we saw before was on the baseline parameter set, but we can simulate different scalings of the simulation tree. And so here I show results for the divergence going from 0.1 up to 1 for each branch. And so before you were seeing data at this column at a divergence of 0.2. And so for each divergence, we train a new neural network and test again specific to that branch of simulation. And yes, I'm sorry, I'm showing the area under the curve for the RSC plots and the precision recall area under the curve. And I've just moved that up to the right column. And then on the left, we can fix the decision thresholds to the one that I mentioned before, and also look at accuracy, true positive and false positive rates, and applied to different aligners. And again, we're seeing that the bold lines are sitting above the dashed lines. And this is particularly pronounced for the higher divergences where PAML basically plateaus to almost a random classifier. So even though the neural network, when trained on Clustau, generalized quite well, we wanted to see what happened when we trained with a different aligner and actually a more accurate aligner. And so the old results when trained with Clustau in the slightly transparent lines and imposed on top of that in bold is when we train with Prank, which is a more phylogeny aware aligner, which makes it more accurate. And the results, I mean, it doesn't do much better or much worse. I mean, there's some cases for Prank alignments where it does a little bit better, but sort of in the middle divergences, it performs worse for the other aligners. And I wonder if this is a little bit to do with it being exposed to less alignment errors and isn't able to deal with them as well. And also having that trade-off of signal in the middle divergence. So you're having more signal because there's more chance for the sequences to evolve, but also a bit more noise from the alignment errors. Okay, and just to quickly compare resource uses for the two methods at test time. So for the 2000 genes that we use for testing, there was a huge time advantage in running with Omega AI versus PAML. If you were just using, or if you were just wanting to test a few genes, this trade-off probably wouldn't be worth it. But if you wanted to scale up to the level of a whole genome scan, where you have the same underlying tree, but thousands of genes that you want to test, it would definitely be beneficial then. And it does come with a memory trade-off. Lastly, I'd like to show you some results that give us a little bit of insight into what the CNN is learning. In the middle, I show you this same simulated sequences colored by their omega values, the red being omega values that are greater than 1. And this is Clustau aligned. In the top is PAML's empirical Bayes estimation of sites that are evolving under an omega of greater than 1. And here in the bottom is a saliency map where the lighter pixels are areas of higher saliency, which are of more importance to the neural network at prediction time. And what we found is that generally higher saliency tended to correlate with the sites that were under positive selection, which is not very surprising. And so if we take saliency as a wish-washy proxy for site-wise detective positive selection, we can make some light comparisons between the two methods for site-wise positive selection. So here, both methods picked up the site under positive selection, and then we can compare a bunch of different sites about when they agreed and when they didn't agree and why that might be. Okay, so to summarize, Omega AI performs favorably compared to PAML when trained for a specific divergence. And this was particularly evident when we looked at very difficult cases where the divergence scaling was very high. And once trained, it is magnitudes faster than PAML. The data that we simulated in this project so far has been of similar complexity to that which is tractable by likelihood methods so far. But through CNNs, we have the ability to simulate increasingly complex data to better mimic real life data above that, which will eventually be too much for likelihood methods as it currently stands. And also, we've shown that Omega AI seems to be able to account for some amount of alignment uncertainty. As for future work, we've begun a collaboration to develop a transformer neural network to take this project to the next level with the people of Phyloforma that you heard about the other day, so Luca and Bastian. And we've heard a lot about the inadequacy of simulated data when it comes to training our machine learning methods. And so I'd really like to look at incorporating some empirical data into the training regime to make it more, to give it more utility in real life examples, and also be able to try different evolutionary models. So for example, selection on synonymous mutations and that kind of thing. So yeah, thank you very much.